morning, sir. Good morning, one and all.
Yeah, please go ahead.
Good morning, one and all. I am Swarupa. I am from regulatory for second year. On behalf of Sri Vishnu College of Pharmacy, I welcome you all to the third day of third national webinar on current rapid growth on vaccine manufacturing and regulation in India, in association with Indian Pharma Association Education Division. Before starting the session, we would like to express our gratitude to our Honorable Director, Dr. Kumar Neemani, to our beloved Principal, Dr. K.S. Nataraj. Now we will have an AV of Sri Vishnu College of Pharmacy. Thank you.
Hello.
ఏవి రావట్లేదమ్మా.
Yes, sir. We are solving it.
Sir, is the PPT is visible, sir?
సార్ ఇక్కడ ఒక 2 minutes కాలేజ్ వీడియో చూపిస్తాను అందరికి, అంటే టు నో.
Okay, fine. Please go ahead.
Yeah.
So we are having small technical issues, sir. We are going to speaker introduction right now.
Okay.
So good morning, one and all. On behalf of management and all, I am going to introduce our today's speaker. I take this opportunity to welcome today's guest speaker, Dr. N. Harishankar Garu. He is a group leader, scientist and director of National Institute of Nutrition in Animal and Facility. So he is going to deliver today's session on animal models in vaccine development. Here, to introduce briefly about himself, it's my pleasure to introduce such eminent personality. He joined ICMR in 1980 and spent initial years in the Food and Drug Toxicology Division of the Institute, where he worked on chemical, nutritional, and toxicological evaluation and unconventional oils. He pursued his master's degree in the year 1988 in Central University of Pondicherry. He moved to National Center for Laboratory Animal Sciences to pursue PhD in zoology and was awarded in 2002. He serves as an advisory committee member in health and nutrition for reputed government bodies like the Department of Science and Technology, DST, Midday Meal Program, and National Disaster Management Authority. He has published 70 papers in reputed national and international journals and 1 book chapter also. He has received 5 international and 6 national awards for his work on small animals. So now I request Mr. N. Harishankar sir to address the participants. So with this brief introduction, I welcome you, sir, to this webinar session.
Thank you. Thank you very much for the nice introduction.
Thank you, sir.
Is the main screen is visible?
Not yet, sir.
Shall I go ahead, sir?
Yes, sir, please. You can share the screen. Nagarjun Reddy, I have given the permission to share the screen.
Already it's open, sir. No.
Sir, no, sir, our screen is not visible.
Sir, is the screen is visible?
No, sir. No, sir.
Now?
Yeah,
Now
now
it's
it is visible, sir.
Now it's okay, sir?
Yes, sir. Yes, sir.
Uh, I did my graduation in Vijayanagaram Maharaja College. Subsequently, I joined National Institute of Nutrition, Hyderabad, and I improved my qualification to PG level, subsequently the PhD in the field of zoology. And I have served the institute for more than 42 years. And I have rendered my services. Throughout my service, I worked in the field of lab animal sciences. I was instrumental in establishing the state-of-art small animal physiology lab in our institute. And I'm also the guide for pharmacology students as well as zoology students in our institute. I'm the nominee for several colleges in Hyderabad, pharmacy colleges like Guru Nanak College of Pharmacy, Avanti, and Anurag College of Pharmacy, and SSJ College of Pharmacy, like that, several are there. And my field of specialization is the preclinical toxicology program. This is our institute prestigious program. And I'm also involved as a supervisor in our regulatory toxicology program of our institute. And I have visited and debated several presentations in the BIRAC as well as RCGM. So dear friends and ladies and gentlemen, today I would like to speak on the animal models in vaccine development. Animal models means we have to select a suitable animal model for the research purpose. And when we say animal models means we cannot go for a large animals. The animal should mimic the human situation, and the animal should be easily propagate, easy to handle, easy to bleed, easy for the drug administration, and also easy to kill or easy to euthanize. So such an animal model we have to select. And the second aspect is nowadays the infections are going up like anything. You might have all experienced the recent pandemic situation, what the entire world faced. For that particular pandemic situation of the COVID-19, several animal models were tested for the vaccine development indigenously. So I'm also involved as an animal supplier to the vaccine development for the indigenously produced vaccines like Covaxin and Covishield. Our institute proudly say that we are the one pioneers in the field of lab animal sciences, supplied the needy animal model to these two organizations in India. So now, today we are going to have some information on the animal models generally available for the vaccine development and the recent observations what we have made. So this is for the beginners or the young flocks of the pharmacy, B Pharmacy and M Pharmacy students, to some extent pharmacy student also. What is an animal model? The animal model is a living organism in which normative biology or the behavior can be studied. And the animal model in which a spontaneous or induced pathological process can be investigated. And the animal model in which the phenomenon in one or more respects resembles the same phenomenon in humans or other species of animal model. That is called the animal model. So these are all several research animal models are available, like right from Daphnia drosophila to zebrafish, as well as the Rattus norvegicus, and the other large animals also. How the animals are good models for the humans, how they were similar to the physiologically in the aspect of the human anatomy. This is what Sir Caspar Wistar studied in the beginning of 1820 or 1830. After he cut open the viscera of the albino rat, he found that the lungs are at the similar same position for the humans like that only, and the heart in the same position of the humans. Liver, kidneys, how they were situated in the rat are almost in the same position in the humans. Intestine part, bladder parts. So it's almost identical to the human anatomy. Then he started demonstrating the rat as an animal model, avoiding the human cadavers for explaining the medical fraternity. And subsequently, the institute is situated in Philadelphia, Vista Research Institute, Philadelphia, has adopted this as an animal model for the research. And the genetical similarities will be 98% and 75% in the case of Drosophila melanogaster. So you have to see the selection of the animal model, and interpretation is very much essential. So the animal model, they should be easy to find out and fast to perform, and the endpoints can be determined at the time of death or survival, or the plus or minus specific symptoms in a group of given animals. And before 1900, animal models were mainly used to study the pathophysiology of the infections only. In 1870 and 80s, a rabbit model of a damaged cardiac wall was in combination with bacteria was studied in the bloodstreams, and it was recognized to encourage endocarditis. During this process, many bacteria were characterized late in the 19th century, and specific staining methods were developed. And in the early 20th century, some species of antibacterial substances were also discovered among the stains developed. So these are the advantages or the things happened during this particular period. So now the selection of the animal model. The selection of the animal model depends on the need of a suitable species of lab animal to study the safety and efficacy of new vaccines, therapeutic molecules, nutraceutical supplementations, and carcinogenesis bioassay testing in the research. So during this process, for the first time in the world, one of the albino rat, as I said earlier, one of the albino rat was brought to the laboratory in USA in 1828 in an experiment on fasting. That albino rat emerged as the first animal, domesticated animal, purely for the scientific reasons, and the existing rat models in the world at present are the descendants or the derived from the same albino rat only. And the selection of an animal model for the vaccine involves physiologic similarities to humans with a respect to the particular application being studied. Relative cost, availability of the animal model, and husbandry practices in the present in the laboratory has to be considered. So such an animal model we should select. So one of the prime most animal model in which now, since we are going to discuss with the students of pharmacy, F-pharmacy, B-pharmacy, therapeutics, pharmacology, analysis, chemistry people, everything. So in the development of the new vaccine or any drug or a new therapeutic molecule, Swiss albino mice were the prime animal model to be used under the phase 1 trials in preclinical toxicology or the regulatory toxicology affairs. The Swiss albino mice, they are the prime models used for the evaluation of any new test compound for its safety, efficacy, and subsequent usage. And the next animal model is the biopsy mice. If your test compound is concerned with the cancer compounds or the anything related to the oncogenic studies, the biopsy models are the ideal models to be studied. If you wanted to study as such the tumorogenic conditions, the disease progression, that means the cancer progression or the regression with the supplement therapy, if you have to use the C57 black 6 mice. This is the best animal model for the oncogenic studies. And once the tumor has been developed in that, you have to develop the vaccines or the therapeutic molecules against that particular tumor can be studied using this as an animal model. And the last one is the nude mice. The mouse without thymus is called nude mice. That means the role of T3, T4 thyroxine in the body will be depleted and the animal will be susceptible for more and more infections. So if you wanted to study any immunodeficiency disorders, for example, you want to develop a vaccine for the human papilloma virus. These particular nude mice derived from the NIH Washington to our institute has been extensively used by the biological Indian immunologicals, where we have used these nude mice as an animal model by using the administrating the vaccine through the olfactory nodules, that means through the nose. And this nude mice is also an ideal model for the cancer development. Subsequently, the treatment with the new therapeutic molecules. Of course, there are several hundreds of the mice models are available. Nowadays, there were some customized animal models are also available for the study, like AKR mice model for the leukemia study. If you want to study you have a test compound developed by you, and you have a test compound or a new therapeutic molecule developed by you, which you wanted to study as for its efficacy or the anti-leukemia studies purpose, you can use this AKR mice as an animal model. Or you wanted to go for a C3H mice where you can study the mammary gland tumor. Nowadays, the breast cancer is alarming in our country, not only in the country, in the world. So the breast cancer, you wanted to develop any therapeutic molecule or a preventive vaccine or any new compounds to be tested, C3H mice, mammary gland tumor models are the best ones. So these are some of the basic mice models for the study of any compound to be tested for its safety efficacy. And now coming to the rat, under the preclinical, one thing I would like to tell you here is, you have developed a test compound, you have developed a new therapeutic molecule, or you have developed a vaccine, whatever it is, first you have to test this for its safety efficacy in rodent models. The mice models, what we have studied, comes under the one rodent model. And if you wanted to study another rodent model, you can go for the Wistar strain of rats. I mentioned here WNIN, that is Wistar NIN rats. That means the rats propagated, born and brought up and propagated at the National Institute of Nutrition. That is why we have named them as a WNIN rats. This is almost similar to the humans populations. Like once a girl getting married from this house going to her in-laws house, her surname will be changed. Once you get a daughter-in-law from the other house to our house, her surname will be changed. Like that, animal models in a particular place crosses more than 24 generations. They will be given the local taxonomy addition to their original name, that is Wistar NIN rats or Wistar albino NIN rats or WNIN rats. They are the best model for the toxicological study. For example, we have done some studies on the CRY1A or CRY1B genes, And we wanted to see the effect of these particular cry1AB proteins and everything. How does they will affects the animal once they introduce it into the cotton gene, cotton product, I mean cotton plantation and everything. There will be any alterations happen to the DNA profile of that particular plant. So we have initially studied its safety efficacy on the same animal model, which are anion rats. And now coming to the SD rats. SD rats are nothing but Sprague and Dolly. The story is the first animal model developed is Vistar albino rat. From there, the colleagues of Sir Caspar Vistar, Dr. Sprague and Dr. Dolly, these two put together isolated a strain from the same rats, and these rats shown that they are the best model for the carbohydrate intolerant mechanism. And they are the best model for the overweight studies. They are the ideal models for high-fat diet-induced nutritional programs. They are also ideal model for the screening of some of the toxicological compounds were involved with the metabolism and diabetic complications. For such studies, Sprague-Dolly is the ideal model. So it is otherwise known as a SD rat. And Vistar Kyoto rat. This Kyoto rat is from Japan. This Kyoto rat has been derived from the cross born and brought up, the Vistar rat born and brought up in Kyoto, Japan, and they named it as a Vistar Kyoto rat. It's useful mainly for the immunodeficiency disorders or the DMSO derived oncogenic studies. Fisher 344 rat. This is the best model for the cancer studies. So you have a test compound or you have a molecule developed as an anti-cancerous agent that can be studied by inducing the tumors in these Fisher 344 rats, and they can be treated with the compounds. So this is about under the rodents, mice and rat models we have studied. Now we are going to have some information on the Mongolian gerbils. Mongolian gerbils are otherwise known as a desert sand rats. They are the best model for the cholesterol mechanism, cholesterol synthesis in the body, the conversion mechanism from the HDL to LDL. Everything can be studied in these gerbils. And they are also ideal model for the skin cancer. So you have a compound for the skin cancer to be treated. So you can create the cancerous situation in these Mongolian gerbils. Though they belong to rodents, they were not called as a rats. They were gerbils. So the tumor formation, generally in our pharmacy colleges, the disease progression means tumor progression and tumor regression will be studied by the volume of the tumor, size of the tumor by vernier calipers. But the time taking from after the induction of the cancer cell lines in the mice models, it may take minimum 21 to 24 days for the gravid tumor formation in the mice. Whereas in gerbils, it comes by 14 to 16 days. So the disease progression will be more faster in these gerbils than the mice. So you have a compound to be tested for anti-cancerous activity. These gerbils can be used. And these are the ones, golden hamsters. Our elderly people will say that don't mix up cabbage and tomato and consume frequently, because that will both the vegetables will have excess calcium oxalate. The excess calcium oxalate consumption will lead to the kidney and gallbladder stones. So what will happen? The bladder rupture will take place, bleeding will occur, death will take place. So to study such situations, we need an animal model, that is hamster. These golden hamsters are the one and only species available in India, that too available in Hyderabad, that too in ICMR, National Institute of Nutrition only. So these hamsters were extensively used for the kidney and gallbladder stones, and these hamsters or cheek pouches are the more prominent one in them. The cheek pouches were extensively used for the tumor formation, like alkaloid extractions or the N-nitrosamine extractions of the tobacco and its products, like Manikchand Gutka, Sagar Gutka, Ambar Kaini, Jodabail Kaini. All these consumption will lead to the oral and submucosal fibrosis will occur in the humans, and gums muscles will be calcified. They cannot open their mouth and the death will occur. To study such situation, these are the golden hamsters extensively used. And these hamsters are also used for the reproductive capacitation test. That was the work done by Dr. Shivaji of CCMB Hyderabad, where he showed the hamster egg cell penetration test will tell you the potential breeding nature of the endangered species or any animal. So that is a program carried out at the Lacones of Hyderabad. So there also these hamsters were extensively used. And these golden hamsters were known as a Syrian hamsters also. They are the one comes under rodent model. So far, my dear friends, so far we have studied mice, rat, gerbils, hamsters. All these four comes under the rodent category under the preclinical toxicology program under phase 1 studies. And if you want to go to the phase 1 study in the development of vaccine or any safety efficacy compound or any compound, you have to go for the second species, non-rodent species like guinea pigs. Guinea pigs are the one extensively used as a second species for the safety efficacy of any compound, and they are also used for the dermal toxicity, like the development of cosmetic products like Revlon, Pond's cold cream, and Odomos, Balsara Odomos. Here I wanted to share that during the 1980-81, we have studied the dermal toxicity nature of Balsara's Odomos at our institute. I was involved as a technical person. And these guinea pigs are also ideal models for the tuberculosis. So, in preclinical toxicology program or the regulatory toxicology affairs or anything under the phase 1, we will be doing rodent models. Under the phase 2, we are going at the non-rodent models like guinea pigs, and the guinea pigs are the one used for serodiagnostic purpose also. Or you have developed a compound vaccine, you have developed a monovalent compound vaccine, you have developed a pentavalent or a bivalent vaccine for the diseases. So what we will be doing is the potency of that particular vaccines will be tested on these guinea pigs only. So the next animal model is the most important prominent one is rabbits. They are the best animal models for the serodiagnostic purposes. You have developed a compound, you wanted to see its efficacy and the serodiagnostic nature of that compound, antibody production, everything will be done using these New Zealand white rabbits. They are extensively used under the phase 2 studies of preclinical toxicology program. Once the small animals like the mice, rat, rodents were over, then large animals, next to the non-rodent models like guinea pig and rabbits were used for these studies. We'll be going to the beagle dogs. You have seen this dog also in a film called Manam, Telugu film by Akine Nagarjuna, Nageswara and his son. Like that, in that film, this dog was there. They are the docile dogs, they are the beagle dogs, the best dog animal model for the second phase of the preclinical toxicology program of the compound what you have developed. So these beagle dogs are also ideal models for the PKPD studies, or you wanted to go for any telemetry studies, non-invasively, you wanted to see the blood pressure and everything with a remote sense. If you want to monitor, these beagle dogs are extensively used. Of course, once the phase 1 trials were over for the test compound or the vaccine, whatever you have developed, then the studies will be done under the phase 2 trials. You have to do it in a species called monkeys. Rhesus monkeys, they are also called as a macaque, and they were extensively used for the preclinical toxicology program under the phase 2 trial. The phase 1 trials were succeeded, then it goes to the phase 2 trials. The compound what we have developed, or the compound what we have developed for the new therapeutic purpose, or the vaccine what we have developed, can be or has to be studied on these macaques only. This is monkeys, because we are all originated from the same blood group RS factor. So then it will goes to the phase 2 trials under the primates. Then once they were also proven safe there, then it goes to the human volunteers. This is the regular phenomena in the regulatory affairs, or the regulatory toxicology, or the preclinical toxicology, whatever you call it. These are the animal models were used. So now we are going to have a small one. What we were also involved in the development of the vaccine. You are all aware that some of our or majority of us were experienced with the corona vaccine either in the first phase or in the second phase, and or in the third phase, where the Omicron, of course, may not be that much serious. Hello? Hello? Shall I go ahead?
Yes, sir. Please go ahead.
So we are all experienced a dreaded disease, or like a pandemic. The entire world is experienced by COVID-19 or the coronavirus. Most of the people lost their lives. Relatives also lost their lives. Kith and kin were lost. And the entire country, entire world has become standstill due to this pandemic, due to COVID-19. So I'm going to have some relevant information on the animal model used for these particular studies. They are: the emergence of new pathogens in humans can impose huge public health and economic burdens on a global scale, in large part because lack of pre-existing immunity. At the end of the 2019, a novel coronavirus, severe acute respiratory syndrome coronavirus 2, SARS CoV-2, that caused respiratory disease and transmits among the humans, was detected in Wuhan, China. Subsequently, it spread to the entire world. On 11th March 2020, the World Health Organization declared that the infections caused by this new coronavirus had reached the pandemic proportions in the world. This is the information what we have. And so how to develop a vaccine for this particular program has been initiated. A key strategy to protect the humans from this dreaded pandemic virus and to develop an effective vaccine and therapeutics was planned. Animal models that closely resemble the pathogenesis of SARS CoV-2 induced disease, coronavirus disease 2019, was planned. In humans, it is essential for the research on disease mechanisms. That means What is the putative mechanism behind the development of this coronavirus infection and rapidly it's spreading to the individuals? That mechanism they wanted to study. And what is the causative agent? What is the spike protein responsible for this one has been evaluated to develop a potential vaccines for this sort of an to develop an antiviral drug for this sort of a virus. It was reported that SARS CoV-2 caused a severe lung disease in hamsters and suggested that hamsters could serve as a useful mammalian model for COVID-19. The pathogenicity was evaluated and the tissue tropism of SARS-CoV-2 isolates was studied in the hamsters after a intranasal infection and found that the virus replicated efficiently in the respiratory tract of these particular hamsters. And they determined that the infectious titer of the virus in only the nasal turbinates and in the lungs, not in the other tissue, which is almost identical to the humans where the COVID infection has been spreading. So the same golden hamster again served as a big tool for the development of the vaccine for this dreaded disease. In general, in the world, there are two types of hamsters are available. One is golden hamster, one is the Chinese hamsters. Though the disease was initially observed in China, Chinese hamsters did not cause this particular infection among the hamsters and they did not develop the infection. However, the golden hamster, the Syrian hamsters otherwise known, developed the same COVID-19 infection of the same virus. The difference between golden hamster and the Chinese hamster is called as a dwarf hamster. There will be two chromosomes depleted from the main hamster. 42 will be there, 40 will be here, chromosome. That is the difference between the two. So now the present discussion will be concentrating on the golden hamsters as an animal model for the COVID-19 infection. So Syrian hamsters as a small animal model for the SARS CoV-2 infection and countermeasure the development. Of course, as I said here, this COVID-19 emerged as a China and it has spread all over the world. Effective vaccines and therapeutics were planned and used. Several vaccines were developed and several people were succeeded in the development of vaccines also like Pfizer people, Moderna people, Sputnik people. So many has been done. But as far as our India is concerned, indigenously developed vaccines like Covaxin and Covishield. Covaxin is developed by the Bharat Biotech Company of Hyderabad. Covishield is the Serum Institute of India, Pune. So now the Covishield and Covaxin vaccines were initially they were tested for their safety efficacy after isolating the spike protein and they were given to the mice and everything rat they were studied, but the maximum help they have got it from these particular Syrian hamsters where the SARS-CoV-2 isolates replicate efficiently in the lungs of the Syrian hamsters and cause severe pathological lesions in the lung of these animals, which are almost identical and similar to the commonly appearing in the humans suffering with this particular COVID-19 infection. And SARS-CoV-2 infected hamsters mounted a neutralizing antibodies response and were protected against the re-challenge with SARS-CoV-2 infection. Moreover, passive transfer of convalescent serum to the hamsters inhibited the virus replication in their lungs. And Syrian hamsters used as a major tool for the evaluation and production of the vaccine immunotherapies of antiviral drugs relevant to this particular COVID-19 infection. Till the time, they were not popular. They were popular only. Hamsters can be used against to the oncogenic studies. Hamsters can be used for the hamster cell penetration test, reproductive capacity test, that's all. But now, these hamsters came into the limelight with the COVID-19 infection, and they saved the entire human population, being an animal model responsible for the development of the vaccine, which we have used very recent past. So the replicative ability of SARS CoV-2 isolates by evaluating the infectious titers in various organs, whether it is in the lungs or the liver or kidneys, in two different age groups of hamsters following inoculation of both the nasal and oral route were studied. But the nasal route proved to be very efficient, and the virus replicated to higher titers in the upper respiratory tract, nasal turbinates, than in the lower respiratory tract of the lung. It was also observed that the virus replicated efficiently in the respiratory tracts of both young and older infected animals. There was no difference in the growth curve of the animals, whether it is upper or lower respiratory tract. But what happened very recent past, we are coming across that the post-COVID scenario leading some of the symptoms which are not seen, but they are causing some drastic changes in the human population. And some people are getting very growth reduction, in the sense body weight loss, and some other things are also happening. And that's a post-COVID scenario. And once the COVID virus was inoculated into the hamsters, the in vivo X-ray micro CT computed tomographic images showed that the progression of the lung inflammation caused by SARS-CoV-2 infection was seen. Subsequently, after the administration of the vaccine, the recovery process was also seen in the same hamsters. So this is the micro-CT images of the lungs of infected Syrian hamsters, which I have taken from a paper published in a reputed journal. And where you can see the lung infections were very much seen, and slowly they are coming down. If you take the initial one, the day 1 to day 14, how they are coming. After treating, this is the lower track one, and the lower intensity and the high dose intensity was also seen in these lungs. So they proved that the infection has been reduced and the hamsters were survived without any mortality. And the same thing was seen in the lung pathological pathological findings were seen. The histopathology examination showed that lungs of the infected hamsters has becoming normal after the supplementation. If you go from the day 3 to day 1, the changes were becoming normal after the supplementation or the induction of the vaccine. So, in COVID-19 patients with acute respiratory illness, the main clinical manifestation is severe lung inflammation. We all know that. Consistent with a previous study using Syrian hamsters, the data demonstrated that SARS-CoV-2 replicates efficiently in the lungs of Syrian hamsters and causes severe pathological lesions of the lungs of these animals. In addition, the micro-CT analysis revealed that the severe lung injury occurs in infected hamsters, and that the severity of lung abnormalities are also related to the degree of infection in a dose-dependent manner. In conclusion, data indicated that hamsters are highly susceptible to infection with SARS-CoV-2 without the need of prior adaptation and development severe pneumonia similar to COVID-19 patients. Importantly, the use of this animal model would facilitate the researchers to evaluate the vaccines, antiviral therapy candidates at a relevantly low cost compared to other animal models such as parrots, and non-human primates, which have also been shown to be successful animal models to SARS CoV-2 infection. That's where in my beginning of my lecture, I told you the selection of the animal model cost effectiveness has to be taken into consideration for the development of any vaccine. In addition to the vast majority of hamsters did not die upon the supplementation with this COVID-2 infection, which is consistent with the human infection. It's interesting to be important to develop COVID-19. Hamsters are the best animal models with low comorbidities such as diabetes mellitus or hypertension or obesity. So Syrian hamster models will be useful for understanding the SARS CoV-2 pathogenesis and testing vaccines and antiviral drugs. So we have supplied to the hamsters to Pune people as well as Hyderabad, Bharat Biotech for the development of the Covaxin and Covishield, and they were successfully released into the market after crossing all the precautionary measures and test process by the regulatories and the DCGO's guidelines, and they were introduced into the human chain. We have also taken a single dose, double dose, booster dose also. We are all surviving. So what I mean to say is, we, the NIN people, National Institute of Nutrition, under the aegis of Indian Council of Medical Research, proudly say that we were also involved in the development of COVID vaccine by participating in the supply of our golden hamsters to them. Thank you. Thank you very much. Hello.
Sir.
Enjoyed my presentation.
And we thoroughly enjoyed our presentation, sir. Really, we are happy to hear this session. Our director, sir, want to speak few words with you, sir. Over to you, sir.
Any questions? It's not audible, sir.
Yeah. Good morning, all the participants. If there are any queries, please drop the questions in the chat box. Sir will respond soon. Sir, over to you, Director, sir.
So, any questions? I think.
Hope you all enjoyed my presentation.
Yes, sir. Yes, sir. A very insightful presentation, sir.
And what I suggest is, as I spoke to you earlier, we should have the one-to-one interaction with the students, and the interaction will give them. And also, we should have planned for a hands-on session workshop to the students, sir.
Yes, sir.
So
yes,
that
we are interested in
what is the mice, what is the rat, how to do the surgery interventions in them. That all can be planned in the near future, sir.
Yes, sir. Ready go. So, yeah. So, sure, sir. Actually, this is what we are planning for, and we want you to visit our facility and also suggest us what we can do. Because a small academic setup, but
no, sir. Vishnu group of institutions are very big.
Yes, sir. Yes, sir. I mean, I will because I see in the NIN facility and CCMB or our ICT or Dr. Reddy's.
Yeah.
I was with Nicholas Permal. I was at Lupin Research Park.
Okay.
We have big research centers, big labs.
Yes.
Relatively, this is very small, but we try to keep some standards and people like you come and visit and encourage the students. It's a great, bless the students. It is a great thing.
Definitely, sir. It's a great honor to us.
Yeah.
And what I mean to say is you can plan for a 1.5 day workshop to the students.
Yes, sir.
Hope you have the air-conditioned seminar halls with you.
Yes, sir. Yes, sir. We have nice facilities and even our animal house is fully air conditioned.
Fantastic.
So,
What I suggest you is, sir, the carotid artery ligation,
yes, sir.
systemic stroke model development.
Yes, sir.
That is one. Carotid, after that, cardiac ligation, heart ligation, and the development of disease model. And
Yes.
oophorectomy, that means overrectomization for the animal model for the overweight syndrome. And also you can go for the adrenalectomy.
Yes, sir.
So these things can be shown to the B-Pharmacy, M-Pharmacy students,
Right, sir.
and they should be given a chance to handle the animals.
Yes, sir.
They should be given a chance to handle the animals with blood drawing, drug administration, and how to handle the animals can be demonstrated to them in person.
Right, sir. Sure, sir. Actually, we are looking for it. As of now, because we are having PG students and almost 25 students will be there at any time. And they are doing projects and we are now developing animal models of Alzheimer's.
Yes.
We are developing these chronic models and looking at memory models like various measures. So we have all the setups with the nice AC facilities.
Yeah, yeah. So, and in the other lines, for some faculty members, can be have a chance to interact with us on the project development.
Yes, sir.
Relevant to the funding agencies. How we
Right.
have to go for it. That also discussion it will come. You can plan for it, sir, because the CMR college, Kandi, Hyderabad people, they are going for a 1.5 day workshop on 25th and 26th of March.
Okay.
So you can also plan for a 1.5 day session there. If your college is well equipped, spacious, you can also ask the other college B students to join them. So
Correct, sir.
they will have a feast of animal science program.
Right, sir. Exactly, sir. That's what we are dreaming to do it. So at this moment, we have close to 200 plus rats in our animal house. And we are getting funding from government, from DST, DBT, and projects are going on. Apart from that, we are also doing consultancy projects for industries.
Fantastic.
We are getting work from industry and we are doing some studies.
What is the role of the students' participation in that? Are they involved?
Yes, sir. The students are the experimenters and they are also co-authors.
That's great. That's great.
Yes, sir. Yes, sir. So they got, but these are at early stages. We are now giving sufficient training and spending time with them.
Fantastic.
Also writing review articles and they are going into the research mode.
Yes.
We have that practice.
In their future, they will be developing interest towards the research.
Right, sir.
After completion of their degree, they need not go for Grama Sachivalaya volunteer.
Right, sir.
They need not go for a call center. All such things can be avoided.
Exactly, sir.
They can be a bridge between the scientists and the management technique.
Perfect, sir. Exactly. That's what we are trying and dreaming to aim our students. And we also have a journal club, sir. Like the all PG students are supposed to present 4 research articles, and they are all highly paid science direct or above science publications. So they present to the all other students and the faculty. So close to 100 papers during PG, they see the research papers.
Sir, second I think Dr. Mahesh,
Yes, sir.
presented the regulatory top.
Right.
Mahesh and myself, we both are involved as a DO for these research methodologies and experimentation part and everything.
Yes, sir.
Do the surgery, we will do the theory aspect, everything.
Perfect, sir.
You can plan it, sir, or else it
Yes.
maybe sometime also you can plan it. Meanwhile, you can have a one-to-one interaction with both of us, to your faculty as well as
Right.
students, sir.
Yes, sir. So we really look forward for the day, sir, and we'll welcome Maheshar and you, and we'll make a workshop and also utilize our own expertise here
Definitely.
to the not only our students and train the students in.
Yes.
So that was the goal. We developed that Center for Preclinical Research Excellency.
Oh, fantastic.
Facility is for that purpose. So we want to train the people fit for doing research and getting into industry.
Thank you. And
And why? Yeah.
thank you very much for the opportunity, sir.
Yes, indeed. Actually, we should thank you, sir. It's our pleasure. I saw I followed whole your presentation, and it's a very exhaustive presentation. And you told me as if I are a professor, sir. So it's so simple that talk. Because I like the way you made it so simple and you so
Yes,
many.
sir. Yes. Because the young class should aware of all these things.
Yes, sir. Yes. So we have in PG that pharmacological screening is one subject. So where we teach all models in depth, sir.
Definitely.
All these models. So we teach them also, and they also now we're trying to practice it in the lab.
Definitely.
And people like you and visitors and motivators and guide us, I think we will go to the next level.
Yes. Thank you.
Really, thank you, sir. Your presentation is wonderful, and we are looking for a day to come to the campus and also do the workshop. And we are trying different techniques, maybe 2-3 days of workshop and seminars. And research methodology and biostatics is also there in the PG program. I teach personally because I like teaching.
And in the NTR University of Health Sciences, I presented some 3-4 times.
Okay.
For the research, MD, MS people, those are doing research, sir.
Yeah. Sure, sir. So we'll make some talk and then also make that into practice doing that workshop model, sir. So we'll design, and I'm looking forward for your interaction.
Thank you.
And I have your number not long back, so we'll be in touch and looking forward. So thanks, sir, for giving this wonderful webinar today.
Right. Nagarjuna Reddy garu, thank you.
Sir, actually today we are very lucky to have a very distinguished speaker like you, sir. And I want to propose a vote of thanks for your wonderful and insightful session regarding this preclinical studies and animal models, sir. And today we had got a very good idea on the how the animal models are introduced in 1828, and later what are the types of mice and what are the types of rodents, what are the guinea pigs, what are the hamsters, what are the uses of using each model, and how we are using the hamsters for the COVID vaccine development. Everything we had a very insightful session, sir, and we are looking forward to many interactive sessions like this in future also. And very heartful thanks to you, sir. That's all from our side, sir. And
Thank you very much.
future, we will be contacting you frequently, sir. Thank you, sir.
Thank you, sir.
Hello.
Yeah, sir, thank you. Yeah, bye.
Yes, sir.
Yeah.
So on behalf of management, our director sir and principal sir of Sri Vishnu College of Pharmacy, we thank all the participants who have participated in 3-day national seminar. Yes, you can leave. So thank you, one and all.